Hello everyone, welcome to another MCAT ACE lecture. In today's lecture, we are going to discuss the recombinant DNA and the biotechnology. This is actually one of my favorite lectures. I'll get a chance to show you how we can use all these techniques to create different transgenic animals for studying human diseases, how we can create a recombinant DNA for production of different proteins, like for instance insulin, for treatment of human diseases, and so on. So this is, in my opinion, one of the very cool lectures that shows us how we can use the advancement in genetics in helping the humanity. Alright, so I'm going to split this lecture in two sections. Part 1, I'm going to discuss gene cloning, different DNA libraries, genomic versus cDNA, discuss how we can perform the polymerase chain reaction through denaturation, re-annealing, and hybridization, discuss the function of restriction enzymes, and then finally go over the gel electrophoresis, southern blotting, and the technique for creation of different transgenic animal models, and then finally how to check for their gene expression, which can be done by different techniques. Like for instance, we can use real-time PCR, we can use southern blotting, we can use northern for RNA, or we can use western for protein, and southern is for DNA. RNA and western for protein. So let's start with the lecture. Let's starting with gene cloning. Can you tell me what does gene cloning mean? It means making many copies of a particular sequence of DNA. So making many copies of a particular sequence of DNA. And for gene cloning, it has to be done through a series of steps. In step 1, a fragment of DNA which contains our gene of interest is cloned into a circular DNA called vector. So here we have the vector, and then we open it up with a restriction enzyme and insert our gene of interest inside the vector. And now this resultant DNA is called recombinant DNA because it's actually being artificially made by combining constituents from different organisms. So here now we have a recombinant DNA. In step number 2, we are going to transfer the vector into the bacterium. So transfer vector into bacterium, and this is usually done by different techniques, but the most commonly used one is electrophoresis, where a short electric pulse is used to create so many holes inside the bacteria, and consequently recombinant DNA can enter the bacterium through all these pores. In step number 3, actually let me pick a different color. In step number 3, we'll let the cell divide. And so one advantage of this recombinant DNA using the plasmid is that plasmids will replicate independent of the nuclear DNA. So once the cell divides, this plasmid will also divide independently of the cell, and now we will have many copies of the plasmid. Many progenies will be made that contain the vector. And so once many divisions are made, a colony of those cells are made that contain the vector, and this vector is containing the recombinant DNA. So now we have so many cells carrying the vector or the plasmid that carries our gene of interest. And so here I've created a drawing for you to summarize all these steps. So there you go. You can see that here we have the human cell. Let's say that in this instance we want to make more insulin protein. So what we do is that we isolate the human DNA and cut the insulin gene with restriction enzymes that are flanking the gene, and I'll go over the function of restriction enzymes later in this lecture. Likewise, we will cut the plasmid open with the restriction enzymes, and now with the help of the ligase enzyme, we will glue gene of interest with the vector. So ligase enzyme helps bind ends of these two DNA fragments together. The resultant vector is called a recombinant DNA. Now via the electrophoresis, we will create holes inside the bacterium so that the vector could be picked up. Now that the vector is picked up, we let the bacterium to grow and produce many different colonies. These new colonies are now carrying the gene of interest. Now you can do it as you wish. You can isolate all the DNA and use the same restriction enzymes to have millions of copies of the gene of interest, which is insulin. Or you can try to isolate the protein from the culture and isolate insulin from the mix of proteins. So there are many things that we can do with it once we clone a gene of interest. Now, there are different forms of DNA libraries that we have, but first of all, what is the definition of DNA library? It's actually a set of DNA fragments that collectively represent the genes of a particular organism. So it's similar to a library. We have many different books that tells us about the knowledge of the humanity. So DNA libraries has a set of DNA fragments that represents the genes of a particular organisms. And it could present in two different forms, genomic and cDNA library. And here, let's go over the difference between these two. So the first DNA library that we have is called Genomic DNA library, and as the name implies, these are the DNA that are isolated from genome of that organism, and so therefore it consists of total chromosomal DNA of an organism. And how it's being done is that we cannot clone the entire DNA into any vector. It's too big. So what we do is that, like, let's say we have a bacterium here, we will isolate the DNA or the chromosome, same thing, and digest it with different restriction enzymes into many small fragments of DNA, and now clone these small fragments into vectors. Let me show it in a different color. That would carry different ones. So now we have many different vectors that each of them are carrying a different DNA fragment, but they collectively represent the entire genome of this bacterium that we've been testing right here. All right, so that's an example of the genomic DNA library. It's a little harder to obtain genomic DNA library because they're larger than cDNA. And here I'm going to explain to you why. So next we have the cDNA library. Can you tell me what cDNA is? So actually cDNA stands for complementary DNA. So once we obtain mRNA, we purify mRNA from a cell. If using the reverse transcriptase enzyme, we create DNA. This is called complementary DNA. And so let me remind you that once we have a gene, it has many different segments of exon, intron, exon, exon, introns. And which of these fragments during the mRNA transcription will be spliced out? The introns will be spliced out. So the mRNA that we are having will no longer have the introns. So the new mRNA will be representing only the exons, and so therefore the protein that is being produced is only from exons. Now for creation of cDNA, we are using this mRNA right here. So there are no introns once we are creating the cDNA, and so therefore it's much shorter than the genomic DNA, and so it gives us the advantage of being easier to be cloned in different vectors and different plasmids. So it's easier to clone cDNA, but then the disadvantage is that it lacks introns. This cDNA doesn't have introns. So if someone is interested, for instance, in studying different splicing pattern or alternative splicing, it cannot be done with the cDNA library. They have to use a genomic library. So now let's go back and discuss genomic DNA cloning in a little more detail. So in the very first step, DNA is isolated and purified from the organism of interest, and then it's digested with the restriction endonucleases. So endonucleases, in order to yield many different small fragments of the DNA that collectively represent the genome of that organism of interest we are working with. Now, regarding the restriction enzymes, basically restriction enzymes are endonucleases that are used by bacteria as a defense mechanism to degrade viral DNA. And since viral DNA is not methylated versus bacterial DNA is methylated, the bacteria endonuclease can tell, oh, this is our DNA, we don't want to digest that one, versus the viral DNA is not methylated. And so therefore, endonuclease is used by bacteria to attack viral DNA and defend themselves against lysis. And how these restriction enzymes work is that they recognize certain sequences and cut DNA in that position. So let me show you an example of a restriction enzyme. Here is another drawing that I have created for you. So there we go. We have different restriction enzymes. Here I'm giving you an example of two of them. One is called EcoR1, which recognizes this specific DNA sequence called GAATTC. So this is in one direction, and then reading in the other direction, it reads in the same order, GAATTC. So both directions is giving us the same DNA sequence, and these are called palindromic sequences, meaning that the nucleic acid sequence on the double-stranded DNA in a 5' to 3' direction reads the same for both strands. And not necessarily all of the restriction enzymes cut at palindromic sequences, but most of them actually do. And so here is an example of two of them where we are getting palindromic sequences. So here the same with the ALU1, you can see we have AGCT in one direction and AGCT exactly in the other one as well, 5' to 3', 5' to 3'. Now let me show you how after cutting these DNAs what happens. So here we have the EcoR1. It's showing you the path of how the enzyme will cut the DNA, as a consequence of which now we have two sticky ends and the DNA is separated from each other. The other enzyme on the other hand is cutting straight, and so therefore it will create blunt ends. Now once we have these sticky ends, so here we have a plasmid, let's say that we cut it, we have a double-stranded DNA plasmid that we cut it with EcoR1. So there we go. So now we have two sticky ends. And if we have our gene of interest that has the same restriction enzyme sites on the both ends, we can also cut it with EcoR1 and create similar sticky ends. Now these two sticky ends are willing to bind to the plasmid. And so if we add ligase enzyme to our mix, it would hasten the binding of DNA fragments to the plasmid and create a recombinant DNA. So here I'm showing you a double strand because I wanted to present the sticky ends to you. And so here again I've created another drawing for you. There you go. I'm showing you how the plasmid has all these restriction sites that can be recognized by our specific restriction enzymes. Cut it open. So this fragment is lost here, right? And we don't need that, that's fine. But it's cut open. The gene of interest has the same restriction enzyme sites. We cut it and create the sticky ends. And now the sticky ends can be brought together with the ligase enzyme and create the recombinant plasmid. Now, if we are working with cDNA, it's so easy. We can clone it right inside the bacterium because it's small enough that can be cloned inside bacteria. But if you are working with genomic DNA, not every plasmid can carry these genomic DNA because they're so large. So we have to use the M13, which is a bacteriophage genome. And so can you tell me what are bacteriophages? These are basically viruses that lyse bacterium. That's why bacteriophage, that's the name given to it. So if you have cDNA, we can clone it inside bacterium, but if it's genomic DNA, we have to use a bacteriophage and a special plasmid for transfer called M13. In any case, so let's say, since we are talking about the genomic DNA cloning, let's say that we have already used the M13 plasmid, and now we have a fragment of our genomic DNA cloned inside this M13. So next, we will infect bacteria with this phage plasmid, as a consequence of which, here let's say that we have a plate and we had the bacteria overlay. Now that we infected all these bacteria with the bacteriophage plasmid, the bacteriophage will cause lysis of the bacteria, and so you will get plaques. Please don't confuse plaques with colonies. If we have a culture plate and we lay bacterium on that, colonies is a bunch of bacterial cells that are growing, versus plaques is once we have an overlay of bacterium and they're being lysed by the bacteriophage. So here, if we have bacteria growing, we call it colonies, while if we have bacteriophage lysing the bacterium, we call it plaques. And so here I have an example for you of the difference between plaque and a colony. I got this slide from Wikipedia. So here you can see that in the first plate we have all these colonies. These are bacteria that are sticking out. So these are colonies from bacterial growth. Versus the other plate, we have the overlay of bacteria. So these are all bacteria that you see right here that are being lysed by the bacteriophages. So here you can see the plaques where there is actually dipping on the plate because the bacteriophages have lysed all the bacteria. So this is an example of a plaque that has been formed by the bacteriophages. So now we have a plate that has many different bacteriophages. Now the next step is to find out which of these bacteriophages is carrying our plasmid of interest that has the gene of interest clone inside, because there are so many plasmids now here and they have picked up so many different DNA fragments, but which one of these is having that gene of interest that we would like to clone in order to understand its function. So next we have to do this screening, and how it can be done is that we can place a nitrocellulose paper on top of this plate, and so once we pick it up, all the different DNA from different bacteriophages will be picked up on the nitrocellulose paper. So now here we have DNA from all these different bacteriophages, and we'll mark this plate. We'll know that, alright, here is the 12 o'clock, for instance, of where we've placed the nitrocellulose paper. Now next we will place the nitrocellulose paper in either high alkali or high salt solution, as a consequence of which the double-stranded DNA under high alkali or under high salt solution will be dissociated into single-stranded DNA. So now all the DNA that is sticking to this nitrocellulose paper are in the single-stranded form. And then in the next step, we will use a DNA probe that has a complementary sequence to our gene of interest. So here is our gene of interest. We will create a DNA sequence that is complementary to one of these segments, doesn't matter which one. And then this DNA sequence is also being labeled with the isotope P32, because DNA, you know how it has a pentose sugar, it has a base, and then the phosphate. So if we use the p32 isotope, it will mark the DNA. So the probe that here we are using is complementary to our gene of interest and is also radioactively labeled with the p32 isotope. So now we will incubate the nitrocellulose paper with our probe, and this probe will bind to the DNA fragments that actually carry the gene of interest. The next step is to place an X-ray film on top of the nitrocellulose paper. And so here, let's say that we have an X-ray film that we've placed on top of this one. And it will show us that, oh, these 3 plaques are the ones that are actually exposing the film and marking the film in these areas. So now we can go back. We know that this was 12 o'clock. We can go back to our original plate and say, oh, okay, so this one, and this one. These were the 3 that are carrying our gene of interest. And so there we go. We've isolated a genomic DNA library that has our gene of interest clone inside. All right, so that was cloning of a gene of interest using genomic DNA library. And once that is done, we just need to confirm that, okay, here is our 3 colonies, but I want to make sure that they have the gene of interest. So next we can use the southern blotting to confirm that they do have our gene of interest. So the next step is to get again another plate. Let's say we isolated 3 colonies. So get 3 plates and have a bacterial overlay, and then infect each one of them with the bacteriophages that we've isolated. So here we had 3 different bacteriophages that were isolated. 1, 2, 3. We are using to create many more copies of the bacteriophage here. Now we can isolate DNA from these bacteriophages. In the next step, we will run them on gel electrophoresis. So here is an gel electrophoresis apparatus where we can load our DNA and let them run. So here we have the negative pole and we have the positive pole there. The DNA has the phosphate backbone. So here I'm giving you an example of a nucleotide. You see how the nucleotide is made of a base, of a pentose sugar, and of a phosphate group. This phosphate group is negative, so the negative backbone of DNA makes DNA attracted towards the positive pole of the electrophoresis apparatus. So all the DNA will be isolated. The larger fragments have difficulty moving through the pores of the agarose gel, while the smaller fragments can easily move through the pores. So the ones that you get on top are larger DNA fragments, and the ones that you get at the bottom are smaller ones. So now that we've isolated the DNA, the next step is to confirm that some of these fragments are actually having our gene of interest. And so here we can use a southern blot, and that I have explained for you a little later. So here we've isolated the DNA on the agarose gel. We will place the gel onto an alkali or salt solution in order again to dissociate the double-stranded DNA into single-stranded DNA. So double-stranded DNA into single-stranded DNA. And we have a stack of paper towel on top and a nitrocellulose paper on top of the gel. So what happens now is that the solution will go through the gel, through the nitrocellulose paper, in order to get absorbed by the stack of paper towels. And so as it's going through, 2 things will happen. 1, single-stranded DNA will double-stranded DNA will dissociate into single-stranded DNA, and second, all the DNA from the gel will be transferred to the nitrocellulose paper. Now we have a nitrocellulose paper that has single-stranded DNA. And now we will hybridize this paper with P32 labeled nucleic acid probe. So similar probe that we have used earlier for marking the plaques that is carrying the gene of interest, we can use the similar probe here because it will bind our gene of interest. So we will use that probe to find out which of these DNA fragments are actually carrying our gene of interest. And so here we can see that after exposure of the nitrocellulose paper to the film, this specific DNA segment has been exposed. So we know that of the 3 plaques that we've isolated, it's only number 2 that is carrying the gene of interest. So it looks like that while we were trying to isolate plaque number 1, plaque number 2, and plaque number 3, plaque number 1 was probably one other one, either one of those. We picked up a wrong one. Similar with plaque number 3. It was probably one other adjacent ones that we couldn't pick up. And so that's the purpose of southern blot, to confirm that the plaques that we think have the gene of interest does actually have the gene of interest. And so you saw that on our southern blot, plaque number 2 did have the gene of interest, and so there we go. Now we have a DNA fragment that is cloned using genomic library. Alright, now moving on to the next topic, which is cloning of the cDNA library. And this step is a little easier, again, because cDNA is not as big as the genomic DNA. And so for cDNA cloning, we usually use the PCR, or polymerase chain reaction. So here what we do is that we just discussed, we have the mRNA isolated from the cell. Using the reverse transcriptase enzyme, we create a cDNA that has all the genes that are being expressed. So again, that's another advantage of genomic DNA, is that it represents all the genes in a genome of a cell, versus cDNA is only the genes that are being expressed, because we are using mRNA to create cDNA. So what if there are some genes that are silent? Those silent genes will not produce mRNA, and so therefore if we are looking for silent genes, we can't use the cDNA library. cDNA library will only give us genes that are being actively expressed. And of course, as we just discussed, introns are also not present in the cDNA library. But again, the ease of cloning makes us use the cDNA library as much as we can. Alright, so now we have the cDNA. We can next, in the next step, amplify the gene of interest, and this can be done by the polymerase chain reaction, or PCR. So PCR is an in vitro technique that amplifies a sample DNA using the DNA polymerase enzyme. In conjugation with a supply of different nucleotides, including adenine, thymine, cytosine, and guanine. And then we have to supply a primer in order to amplify a region of interest. And here I'm going to go over that with you. So we had the mRNA, we used the reverse transcription to create cDNA. So now we have the cDNA, and let's say that our gene of interest is right here. How can we amplify this gene of interest? So if we know the sequence of the surrounding of the gene, what we can do is that we can produce single-stranded DNAs that are complementary to these flanking regions. And so these are called primers. Primer. And then we have also nucleotides like adenine, guanine, cytosine, and thymine, and we have DNA polymerase. So I'd like you to remember these components that we have: primer, nucleotides, and DNA polymerase. Now let's explain how the PCR reaction will help us amplify the gene of interest. Alright, so now in the first step of the PCR reaction, we will denature the double-stranded DNA into single strands. So denature double-stranded DNA into single-stranded DNA by heating the solution to 98 degrees centigrade. So now here was our cDNA with gene of interest that was right here. It's now going to split into two. And these are all single-stranded DNA molecules. In the next step, so this was 98 degrees C. In the next step, we will lower the temperature to 64 degrees C in order to allow annealing to take place. And annealing is when the DNA primer attaches to single-stranded DNA. So primer attaches to single-stranded DNA. So now at 64 degrees C, annealing can happen. So here we got our single-stranded DNA. Given that the temperature is now appropriate for the primers to bind, the two primers will bind to the single-stranded DNA. And now in the next step, the third step, we will increase the temperature to 72 degrees C, which is suitable for the DNA polymerase in order to add DNA sequences. So here, the third step is called extension. And so now during the extension phase, from 5' to 3' direction, nucleotides will be added, 5' to 3' direction. So now you can see that through one PCR reaction, a single double-stranded DNA has yielded two double-stranded DNA that is carrying the gene of interest. And now in the next stage, we will repeat the denaturation. So now we will isolate these two double-stranded DNA into 4 single-stranded DNA. Then lower the temperature to 64 degrees C to allow the primer again to bind to them, and then increase the temperature to 72 degrees C in order to add the nucleotides. So now by the second PCR reaction, we have 4 double-stranded DNA. Alright, so through every PCR reaction, the number of DNA that we have will double. And so very fast, after like 10-20 reactions, you will notice that you will have very large number of the DNA that you've started with. And this is basically the approach that is used in forensic studies. Like for instance, you isolate a hair or very small amount of DNA left behind, and then you use the PCR to amplify and make millions of that specific DNA. And now you can analyze it, see, all right, who does this DNA belong to? And I will discuss that in our second lecture. Don't worry about it. And so through the PCR, we can amplify the number of DNA strands that we are having. And so here again, I have another drawing for you that will show you all these steps that we just discussed. So there we go. We have the nucleotides, we have a primer, we have the template DNA with the gene of interest that we would like to amplify, and we also have the polymerase enzyme. We denature the double-stranded DNA into single-stranded DNA at 98 degrees C. During the annealing stage, 64 degrees C, the primers will bind, and then at 72 degrees C, the nucleotides will be added. And so here, a single double-stranded DNA has yielded 2 double-stranded DNA. And now cycle repeats. Again, try 98 degrees C, 64 degrees C, 72 degrees C, and now we have 4 double-stranded DNA. So now that we have many copies of our gene of interest, we can clone it into the vector that is special for E. coli and then express the gene in the E. coli. So the type of vectors that is used for E. coli is different than the bacteriophage. And here we have the advantage of including a antibiotic resistance gene, like for instance ampicillin in this instance. And so only the bacteria that are carrying this plasmid will survive in the presence of ampicillin, because this gene can break down ampicillin. And then we have this other gene called LAC-Z, which expresses the beta-galactosidase enzyme. And in the presence of the X-gal, which is an organic compound, bacteria that are producing LAC-Z will produce blue color. So keep that information in mind. One, we have the ampicillin resistance gene, and second, we have the LAC-Z on the original plasmid. And I'll explain for you why these are important. All right, so just like I explained for M13 vector, what we do is that with the EcoR1, we will use the digestive enzyme to open up the plasmid. And then we will cut the gene of interest with the same restriction enzyme. So here, you know that we have so many products of double-stranded DNA, all right, that have the gene of interest there. Once we are designing the primer, we try to include the restriction enzyme sites, like for instance EcoR1, in our primer. So all these genes are now being flanked by EcoR1 fragments. And so if you add an EcoR1 enzyme, restriction enzyme to it, you will get all the pieces of DNA that has the gene of interest cut out. So now we have millions of these pieces that are added to millions of these vectors that we are having. With the help of the ligase enzyme, you help anneal them together. Now, one problem may happen here, and it's that before the gene of interest has time to get inserted here, the plasmid may close on itself. And so we can have two different scenarios. One is that we will have the original plasmid, or second, we could have the gene of interest, and that's actually what we are interested in. We are only wanting bacteria that are carrying our gene of interest. All right, so we've done all these experiments, we've created the plasmids. Now we are using the electroporation technique in order to make some pores inside the cell membrane of the bacterium, so that all these DNA plasmids can be picked up. And electroporation is done by a short electric pulse. All right, so now we can have different scenarios. Let's go through them. Okay, let's pick a different color. The first scenario is that bacteria may not pick up any plasmid whatsoever. All right, and so what happens here? Our culture plate has ampicillin added to that. So anyone who doesn't pick up this plasmid, they don't have the resistance gene to ampicillin. So they will all die. So any bacteria that doesn't pick up our plasmid will not show up on our plate, because ampicillin will kill them. The ones that show up already have the ampicillin resistance gene. So now the ones that have ampicillin resistant gene, there could again be two scenarios for them. One, do they have the plasmid that does not have the gene of interest? So no gene of interest. All right, and if that happens, you can see that the LAC-Z gene is intact. So in this scenario, the LAC-Z gene is intact, so therefore beta-galactosidase will be produced, which in the presence of the X-gal organic compound, it will produce a blue color. So any colonies here that are having blue color is because their LAC-Z gene is intact. So all these are garbage because they didn't pick up the gene of interest. On the other hand, if they picked up the gene of interest, then the LAC-Z gene is interrupted, and so therefore no more blue colonies will be produced. So all these white colonies are the ones that, one, are carrying the ampicillin gene, and so they survived, and 2, they disrupted the laxi gene, and that's why they're having the white color. So now we can isolate all these colonies and test to make sure that they do have the gene of interest. Again, we can use southern blot for that, or we can use western blot to make sure that the protein that we want is being expressed. Right now here I'm showing you an example of different colonies. So you can here see that we got all these blue colonies here, and these have picked up the plasmid that doesn't have the gene of interest, so we don't need them. But all these white ones are the ones that we are interested in because they have picked up the gene of interest. So that was the technique of cloning gene inside E. coli. Now next I'd like to discuss how we can clone genes in higher organisms, like for instance in fruit flies, and then later on in mice. Alright, so just to provide some background information before I explain cloning in fruit flies. So there are certain DNA sequences called transposons. And these transposons are common in most organisms. They are short pieces of DNA that are usually less than 10 kilobases. And these DNA fragments, in the presence of the transposase enzyme, they can come out of the chromosome and jump around, move somewhere else. So here, let's say that we have a chromosome here and we have transposon sequences right here. Now in the presence of the transposase enzyme, these transposons can come off. So this DNA fragment can come off and move somewhere else in the genome. So they would jump around. So we can use this technology in order to introduce a gene of interest that is flanked by 2 transposon sequences in order to make the gene jump out and insert itself somewhere inside the genome of the animal of interest that we are working with. So here, let's give an example in how we can do that in flies. So transposons in fruit flies, we use a special transposons called P elements. So these are the same as transposons. And so you can see that we have the gene of interest that is inserted in between the 2 transposons. In addition, we have a marker right here. So you know how in E. coli we use the blue color as a marker? We need some sort of marker in flies too, because you don't want to screen like thousands of flies to find out which one of them is actually carrying the gene of interest. So here we are using the W+ gene, which expresses red eye color. Alright, so just keep that in mind. And then we have another helper plasmid that has the transposase gene. So let me ask you a question. Why don't we insert the transposase inside the same plasmid right here? Why should we insert it separately? And the reason for that is that we just want one-time insertion. We don't want these DNA sequences to keep jumping around and cause mutation. We just want it to insert once and we're done with it. So therefore we have the transposase on a separate plasmid in order to express temporary this protein and do its job. And then after that we are done. We don't need it anymore. So there we go. Now we have the transformation plasmid and then the transposase gene. They're all being injected under microscope into the germline cells. So here at the back of the egg are where the germline cells are. And germline cells are cells that will develop into testicles or ovaries for production of sperm or egg. Alright, so the other point I'd like you to notice here is that the egg that we are actually injecting has the W- gene. It means the gene required for red eye is not being expressed. And so therefore the fly that will develop here will have white eye. Alright? So now we have a white eye fly that some of the germline cells are carrying the gene of interest. So these germline cells are carrying the gene of interest. But then there will be also some other germline cells that don't carry the gene of interest because the transposase gene and the transposons didn't insert into their genome. Now from here on we will mate this fly with another fly of white eye color and check their progenies. Any progeny that has white color eye means that they didn't pick up the gene, alright, so we toss them away. But the ones that have red eye, they have picked up the gene of interest as well as the W plus gene. So now here we have a fly that has the gene W plus and the gene of interest cloned inside. Now we can use this fly for many different studies, and I'm just going to give you some example how amazing this technology is. So I've completed my PhD studies in creating different fruit fly models of human diseases, and I personally created Menkes disease, which is a copper deficiency disease. So we knocked out the gene that is required for copper absorption. And once we've created this fly, then we tested expression of different genes, see which one can help these flies survive longer. And so once we've identified the gene, we published our results. About 4 to 5 years later, scientists started to show that, oh, by the way, this gene is protective in Menkes disease. And like, for instance, they've shown the same studies in mice, and then they applied the findings in humans. So my point is that with a simpler animal model system, we can find out genes that are protective against certain diseases, and then from there on apply it to human diseases. Now there are other cool animal models of human diseases. Let's talk about fruit fly, for instance, that I'm sure you'll find interesting. There are actually fruit fly models of Alzheimer's disease. And so basically the protein that is causing Alzheimer's disease is the amyloid beta 42. So this protein aggregates will cause degeneration of cells in the brain of humans and cause Alzheimer's disease. So same gene has been expressed in fruit fly brain, and it has been shown that these flies are now forgetting faster. But how do we check in flies that they're forgetting faster? So here we have a fly, and you expose them to odor A with no shock, and then you expose them to odor B with an electric shock. And then you provide them with a T-tunnel. You let flies go through the T-tunnel. Now they have the option to go through either path, but one of the path has the smell of odor A, the other one has smell of odor B. So normal flies remember that odor B had a shock, so it's not a good idea to go this way, so they'll go towards odor A. Versus Alzheimer's flies don't remember, so it's 50-50. Half of them will go towards odor B and half goes towards odor A. So now that we have a fly with Alzheimer's disease, we can now check different drugs or different gene expression or knock out different genes, see which one can help lessen the severity of disease in these flies. And so it's an efficient way of screening for different drugs. Now let me show you another example. There is also a fruit fly model for Parkinson's disease. And there are different models. There are some that are being done by expression of alpha amyloid protein in their brain, or there are some other models that has been created by knockout of Parkin gene. In any case, it has been shown that these Parkinson flies have similar features to human Parkinson's disease. They're kind of like shaking, and they have difficulty climbing, and they die at an early age. And so later on, these fly models have been used to screen for genes that are protective against Parkinson, and there has been some novel findings of some of the protective mechanisms against Parkinson. So that's how we can help with the medicine by creating all these different animal models. And usually the simpler animal models like flies are cheaper than mice, and so that's why scientists are first trying to create them in Drosophila before they move on to mice. But then the advantage of creating animal models in mice is that they're mammals, and so they're closer to humans, so it's very likely that the findings you find here could also apply to humans. Alright, so here in the next stage I'd like to describe how we can create transgenic mice. And so there are two different techniques that we can use for inserting DNA into progenies. One is by altering the embryonic stem cells, or two, by injecting the parental germline cells, just as we did like in Drosophila, and I explained. So anyway, let's explain first the embryonic stem cell technique. So here we have the embryonic stem cells, and we try to target the gene of interest into those embryonic cells. We isolate the one that have picked up the gene and multiply them, right? So now we have the transformed cells. In the next stage, we will inject the transformed cells into the blastocyst of an embryo, and now we will implant the embryo inside foster mothers. So the mother now has a fetus that some of its cells is carrying the gene of interest. So finally gives birth to the animal that we've genetically created, and this animal is called chimeric because some of them are carrying the genotype of interest and some of them are carrying the initial genotype, the original genotype. So again, we have a chimera, which is a single organism that is composed of different genotypes. All right, so these pink ones represent our gene of interest. Now, some of the germline cells also are being transformed in this animal model. So once you mate our chimeric mouse with a normal one, some of our progenies will pick up the gene of interest, and now their whole body will carry the gene of interest. All right, so again, we have all these germline cells that are carrying the gene of interest, and there are some that are not carrying the gene of interest. If the ones that are not carrying the gene of interest give rise to a progeny, then none of these cells will have our transformed gene. But then the ones that do carry the gene, if they give rise to a progeny, then the entire cells of this animal will now be transformed. So this is no longer, none of these are chimeric because they all have a uniform genotype. Alright, so that's the first technique, using the embryonic stem cells. The second technique is similar to what I just described for you in Drosophila. You basically inject the transformation vector into the germline cells. And now these germline cells, some of them are being transformed and they give rise to animals that are carrying the gene of interest. And the ones that are not transformed, those germline cells that are not transformed, will give rise to animals that are not carrying the gene of interest. All right. Now, the next step is to find out if our gene of interest is being expressed or not. And we have already briefly touched on it, on how to use Southern blot technique in order to see if they are being expressed or not. And so here I'm going to explain that for you again. So next, we can isolate DNA. So let's say here, we have a mouse that we think does carry our gene of interest. So now we can isolate DNA from this mouse, load the DNA onto agarose gel, and so run it through. So now we get many different fragments of DNA right here. All right. Then again, using the apparatus that I just explained for you, either alkali or salt solution, in order to make a double-stranded DNA into single-stranded DNA, we let the fluid to flow through the gel into the nitrocellulose paper, into the stack of a paper towel. And so there we go. All the single-stranded DNA are now transferred to the nitrocellulose paper. Next, we will hybridize the nitrocellulose paper with our P32 labeled nucleic acid that represents our gene of interest, and then visualize the bands on the X-ray film. And so there we go. We can tell that, oh, this one, the second lane is carrying the gene of interest. So whichever mouse, whichever DNA that this lane is coming from is actually successfully transformed. So this technique is called Southern blotting if we use DNA. If we use the same technique on RNA, then it's called Northern blot. So instead of DNA, if we load RNA, then it would be called Northern blot. All right, and some scientists actually use the intensity of the exposed film to say, all right, how much of the gene is being expressed. So here, let's say that they say that this is our control animal, and there is a faint band of the gene of interest. And then here we have our transgenic animal that has a very thick band meaning that the exposed film had so much of the gene of interest. And so this way you can compare by scanning the films and seeing how many pixels is in one compared to the other one. You can compare the relative gene expression. But then if you're using certain genes that is not present in that animal, like for instance in this instance the other two animals don't have the gene of interest anymore, then we can't really compare it for quantification. All we can say is that, oh, the gene, the external gene is present, or the exogenous gene is present. Now, another way to confirm that our experiment has been successful is to use Western blot. And this is actually preferred over Southern or Northern blot, because with those techniques we just confirm that mRNA or DNA is present. And so if DNA is present, how do we know that it's being successfully transcribed and then successfully translated into protein? Same with Northern blot. We can say that, oh, mRNA is there, but how do you know that a successful translation will happen? What if this mRNA is so unstable that no protein will be created? And so our animal model could be useless. And so that's why most scientists prefer Western blot, which checks to see if the protein of interest is actually being expressed. And so next I'd like to explain how Western blot is being performed. So let me clear the board here for you. So we run the Western blot on either a native page gel electrophoresis technique, which separates the proteins. These are undenatured proteins based on their charge and size. And then if we only want to isolate them based on their size, what we can use is that we can use the SDS page gel. And SDS contains this compound called sodium dodecyl sulfite. And so what this compound does is that if here is our three-dimensional protein that we are having, and that's its tertiary structure, what this SDS does is that it has a negative charge, so it will bind to this protein and open it up. So there we go, it will open it up with all these negative charges. And so once we run the proteins on an agarose gel, they would be isolated only based on size and no longer based on charge. So if we use the native page protein, native page gel, proteins will be separated based on charge and size difference, while if we are using the SDS page, they would be only isolated based on size. And so here again I have another drawing for you to show you how the SDS page works. So here we have the native protein that has been used in the native page gel, but then if we use SDS, it will open up the protein because of all these negative charges don't allow the tertiary conformation to be maintained. And so now we can isolate the proteins based on their size. Alright, so now let's describe the Western blotting technique. So here is the Western blotting technique. So what we do is that we isolate protein from our different samples that we are having and load them into an SDS page and run it through the gel electrophoresis. So now we have the negative charge and positive here. We know that all our proteins here are carrying the negative charge. So they will be repelled from the negative side and move towards the positive side. The larger protein fragments will stay higher because they have difficulty to go through the gel pores, and the smaller ones will move faster down towards the bottom of the gel. In the next stage, we are using this gel electrophoresis apparatus, which will electrophoretically transfer proteins into the membrane. And usually the membrane that we are using is called PVDF. It's different than nitrocellulose paper that we are using for southern and northern blot. Basically, this PVDF stands for polyvinylidene difluoride, and it's pretty good for binding of proteins. So now here we have all these proteins with a negative charge that are on the gel, and so again we are running from negative side to the positive side, so they would be transferred to the PVD membrane. So now here we have a PVD membrane that has all the proteins attached to it. So in this next stage, we will use some neutral protein in order to block the membrane, so all the pores will be filled by some native protein, like for instance BSA or milk casein. Alright, and then in the next stage, we will incubate the membrane with a primary antibody. And I'm going to explain for you what's the difference between primary antibody and secondary antibody that we are using right here. So once we are having a protein, in order to create antibody for that, what we can do is that we can inject it into an animal, like let's say we inject it into mouse, and mouse immune system will create antibodies that will attack this protein. So there we go. Now mouse has created antibodies that are attacking this protein. And we can purify this antibody. So now we have a primary antibody that is binding to our protein of interest. So there are some of areas on this PVDF membrane that has protein of interest. So all these antibodies will bind to them. Alright, and this is called primary antibody. And don't worry about the structure of antibodies. We have extensively described the heavy chain and light chain in our immunology lecture. Alright, so here the primary antibody binds to the protein. Now we need a secondary antibody. And so this secondary antibody is actually carrying a chemiluminescent probe. So now we need the secondary antibody to bind the primary antibody. And how we create that is that we have the primary antibody. Now inject this primary antibody into a different animal. So let's say horse. And a horse immune system will now create antibodies that are attacking the mouse antibody. Alright, so this is called secondary antibody. So the secondary antibody that we have now created has a special agent that will have chemiluminescent properties. So this antibody will now go and bind to our primary antibody, and it has the chemiluminescent properties. And so once you place a film on top of the PVDF paper, now because of this chemiluminescent property, the film will be exposed. And so any fragment that will show up on the film means that the primary and secondary antibody has bound to it. And so here you can say that, oh, our second lane, and here let me do it again, our second lane and then the second lane and the fourth lane, these two samples actually contain the gene of interest and are expressing the protein. We just confirmed that the protein of interest has been translated right here. Alright, and then the final technique that I'd like to describe for you, and after that we will do some sample question and conclude the first part of this lecture, is called quantitative real-time PCR. And so basically we are using the same concept as PCR to amplify the DNA fragment, and the purpose of quantitative real-time PCR is to quantify the level of gene expression. So with the real-time PCR, we have a dye that gives the fluorescent signal once it binds to double-stranded DNA. So if you have a sample that has so many double-stranded DNA, we have a strong fluorescent signal. While if you have a sample that has a few double-stranded DNA, there would be a weak fluorescent. And so there is a machine that will detect the level of fluorescent, and this way it will tell us that, oh, this sample has few DNA versus the second sample has a lot of DNA. Alright, so let's say that we have animal A and animal B. Animal B, we've place it under some stressful condition, and we want to see if for instance let's say steroid gene is being overexpressed or not. So what we can do is that we find the steroid gene sequence and create primers that are flanking the steroid gene. So now from both of these animals we isolate mRNA So this is for animal A, this is for, subject A and subject B, mRNA, and then using reverse transcriptase, create cDNA. All right. Now the number of these cDNA could be different between the two of them because subject B has been exposed to stressful condition, and we want to know if stressful condition affect the expression of the steroid protein. All right. So now from here on, we will use the real-time PCR to amplify these DNA fragments, and so we will get a graph that has, so the machine will detect the fluorescence signal from each of these samples. It tells us that sample A, for instance, it took 30 cycles for the sample in order to reach its peak, versus for sample B, it only took 25 cycles. 25 cycles for the fluorescein to reach its peak. So now here we can see that sample A took 30 PCR reactions while sample A took 25. So what's the difference between the two? 5 cycles. So as much as 5 cycles, sample B has more DNA. So 2 to the power 5. So it's 32 times more mRNA of the steroid has been expressed under stressful condition. All right, so that's one way of calculating the mRNA level. But again, we always prefer Western blot, but most of the time we don't have the antibody, because recall, for the Western blot we need the primary and secondary antibody. If we don't have those, we can't really carry out the Western blot, and so that's why most scientists just use the real-time PCR as a quick way of quantifying mRNA. So mRNA gives cDNA, and then here you can compare different samples. Now here, let me give you another example. So here we have the relative fluorescence. Here is our cycles of PCR, 10, 20, 30, and 40. Let's say that sample A took 20 cycles to reach its peak fluorescein, while sample B took 30. What's the difference between these two? Which of these are expressing the gene of interest at a higher rate? Sample A has the gene of interest expressed much higher because it only takes a fewer reactions. It only takes 20 reactions to reach the peak, while sample B, it takes much longer many more reactions to reach its peak. So there is 2 to the power 10 difference between the gene expression. So gene A has 2 to the 10 power higher gene expression compared to gene B. Now let's say we have another one called gene C, sample C, I'm sorry, not gene C, sample C, that takes 18 reactions to reach its peak. So compare sample C to sample A for me. Which one is expressing the gene of interest at a higher rate? Sample C, of course, because it takes 18 reactions to reach the peak, versus sample A takes 20 PCR reactions. So comparing A to C, 2 PCR reactions takes longer for A to be equivalent to C. So sample C has 4 times higher expression of the gene of interest that here we are amplifying using the real-time PCR. All right, and so that's about it. We are done with the first part of this lecture. We've already explained gene cloning, how we can do it with DNA library of Genomic and cDNA. We explained different reactions of PCR, denaturation, re-annealing, and hybridization. We described the function of the restriction enzymes. We described southern blot, real-time PCR, northern and western blot, and then we've also discussed how to create different transgenic animals like mice or fruit flies. All right, now here I'd like to do some sample questions, and then after that we move on to the second lecture. So if you give me a second, I'm going to clear the board and come up with some practice questions. All right, there we go. We have 4 questions to practice right here. If you need time to go through them, please by all means go ahead and pause the video. The first one is, which of the following is not a component of polymerase chain reaction? Which of the following correctly labels this extension stage of the polymerase chain reaction? And then question 3, which of the following DNA probes is best suitable for hybridizing this specific nucleotide sequence? And then the last one is, which of the following DNA sequences could be a restriction site? All right, so if you need more time, you can pause the video and then start once you feel you're ready. All right, the first question. Which of the following is not a component of the PCR reaction? So without looking at the answers, we recall that we needed a polymerase enzyme, we needed a DNA template in order to amplify the gene of interest, we needed some primers, and we needed some nucleotides. So here, primers, nucleotides, DNA polymerase, restriction enzymes is not required for PCR. We can use it later on to cut the gene of interest out, but then it's not required for the PCR reaction itself. Next question. Which of the following correctly labels the extension stage of the polymerase chain reaction? So again, what happens at 94 or 98 degree? This is the denaturation stage, where the double-stranded DNA will be converted into single-stranded DNA. Then we lower the temperature to 64 degree. This is the re-annealing stage in order for probe to bind to the DNA template. And then at higher degrees, 72 degree, this is the extension stage, where the nucleotides will be added in front of the primer. And then we repeat the cycle. Again, we increase the temperature to 94 or 98, which is for denaturation, and repeat the cycle. There we go, annealing, extension, and so on. So the question is, which of the following correctly labels the extension stage? So it would be choice C, is marking the extension stage of the PCR reaction. All right, next question. Which of the following DNA probes is best suited to hybridize this nucleotide sequence? So here we have a 5' to 3'. In order to hybridize, we need, let me show it with a different color. We need a probe that is in 5' to 3' in the opposite direction. Okay, but here, pay attention that we are asking which of the following DNA probes. So any of these sequences that has uracil, these are not DNA probes, these are RNA probes, because remember, with DNA we have thymidine, while with the RNA we have uracil. So choice A and choice B are already out because we know that they are not DNA probes, they are RNA probes. Now, of the other 2 choices, let's see. So here we have GUACUA. If we want in a 5' to 3' direction, we will get TAGTAC. Without looking at the answers, I wrote this down. So let's see. We have 5' to 3' TAG TAC, TAG TAC. So this is 5' to 3'. So 5' to 3' is TAG TAC. So there we go. Choice C is the correct answer, and choice D is incorrect. We don't want CAT GAT. That's 3' to 5' direction. No, that won't work. All right, there we go. Last question. Which of the following DNA sequences could be a restriction site? So basically we discussed that most of the restriction site, not necessarily all of them, but most of them are palindromic, means that 5' to 3' direction, in both directions we get the same sequences. So let's see, for choice A, GGATCC reads the same as GGATCC, so sure, could be possibly a restriction site. The rest of them, however, are not palindromic, and so therefore are not likely to be a restriction site. And so that's about it. That concludes our first part of the recombinant DNA technology lecture, and I will see you all for the second part. Thank you all for listening.